eDNA Elucidates Genetic Diversity and Population Structure by Will Seehaver. Biodiversity is vital for maintaining ecosystem function. Unfortunately, biodiversity is declining worldwide. Furthermore, biodiversity is related to genetic diversity and species richness. Yet, biodiversity in aquatic environments can be very difficult to quantify through capture and similar methods. Environmental DNA, eDNA, allows researchers to quantify the genetic diversity of an ecosystem and determine the types of species present, which has important implications for conservation biology. The study sought to determine whether eDNA can be used to detect and quantify within species nuclear genetic diversity and population structure in natural aquatic environments. Specifically, the study was designed to answer the questions: can nuclear DNA be amplified and analyzed from environmental samples? And does the genetic information from environmental DNA match that from DNA testing of tissue samples? The researchers collected eDNA and tissue samples from 14 sites across the Great Lakes. The Great Lakes are among the most heavily invaded ecosystems in the world. More than 188 non-local species have been documented. Skin cells, hair, scales, feces, mucus, gametes, or decaying tissue can all contribute to DNA to an environmental sample. qPCR was used to quantify mitochondrial DNA, mtDNA, and nuclear DNA, new-DNA, in eDNA samples. There is typically more mitochondrial DNA than nuclear DNA, since cells contain only one nucleus but many mitochondria. Custom primers and TACMAN probes targeted specific gene regions. A panel of 28 nuclear microsatellite loci was amplified from both eDNA and tissue samples. Quality filtering, demultiplexing, and allele assignment were performed. Lastly, allele frequencies, genetic diversity, and population structure were compared between eDNA and tissue datasets using statistical analysis. The study found that eDNA detected 92% of nuclear markers. 66.8% of tissue-detected alleles and 83.7% of common alleles were recovered in eDNA samples. The amount of mitochondrial DNA in each sample was positively associated with the amount of nuclear DNA in each sample. The ratio of mitochondrial DNA to nuclear DNA was 196 to 1. They also found that eDNA-derived allele frequencies showed a strong correlation with those from tissue samples. eDNA allele frequency distances, AFDs, were positively correlated with tissue-based AFDs. However, eDNA had a lower allelic richness and heterozygosity than tissue samples. Additionally, some alleles were overrepresented and some rare variants of alleles were underrepresented. In conclusion, eDNA can be used to reliably detect nuclear genetic variation and species presence. While mitochondrial eDNA is more abundant, nuclear eDNA still provides meaningful population-level insights. Genetic patterns derived from eDNA closely mirrored those from conventional tissue sampling. These results indicate eDNA can be used as a non-invasive method for species monitoring, finding rare or endangered species, and sampling hard-to-assay aquatic environments. eDNA can quantify the genetic diversity of a species, allowing conservation biologists to determine population viability and resilience, inbreeding and genetic bottlenecks, and source identification and movement patterns. eDNA is useful for studying genetic diversity when tissue sampling is not practical, such as for large marine mammals or endangered or vulnerable species. While this study focused on a single species, eDNA samples contain genetic information on multitudinous species, allowing scientists to develop a more comprehensive picture of the species present in an environment and providing the opportunity to examine multiple species contemporaneously. It is an important tool that allows conservation biologists to manage distinct populations, design protected areas, and biomonitor conservation success.